Okay, so we just talked about our definitions then of a solute, of a solvent, and then we talked about the concentration gradient, meaning the concentration of the solute, an area of high concentration moving to an area of low concentration, or only with the use of energy, a solute moving from an area of low concentration to an area of high concentration. So now we're going to talk about the different types of transport. So we start out by saying there's passive transport, which does not require energy, and active transport, which does require energy. So the first type of passive transport we will talk about is diffusion. And remember, there are two different types of diffusion. There's simple diffusion and there's facilitated diffusion. And simple diffusion is, as its name sounds, is more simple. It's just the movement of molecules going from an area of high concentration to an area of low concentration through the plasma membrane. In facilitated diffusion, we are facilitated by one of those intermembrane proteins or transmembrane or integral proteins that are embedded in that plasma membrane. So that protein is facilitating the movement of the molecules, of the solutes, from the area of high concentration to the area of low concentration. So what we see is that diffusion on a whole, in both simple diffusion and facilitated diffusion, are solutes moving from an area of high concentration to an area of low concentration. So that's the same with diffusion as a whole, or between simple and facilitated. The other thing that's the same between the two is that it does not use energy. It's a type of passive transport. So both types, simple diffusion and facilitated, even though they're using a protein in the second one, it does not require the use of energy. So you can see here on the left-hand side, that here we have our extracellular fluid, so on the outside of the cell, and here we have the inside of the cell. So our high concentration of our solutes, some ion or molecule or something, is able to pass directly through the plasma membrane without the assistance of any other molecule. You can see there are more of these molecules over here, ions or whatever they might be, molecules, over on this side than there are on this side. So this is an area of high concentration moving to an area of low concentration, so moving down its concentration gradient. Over here we see the same type of concentration gradient, so an area of high concentration of molecules and an area of low concentration of molecules. But here we see is our protein that we can use for transport, so the one that is facilitating this type of diffusion. So these are much larger molecules, so molecules that have to use a protein to move across the membrane. So it's just with the help of this carrier molecule or transport molecule. So again, passive transport without energy, and diffusion is our first type of passive transport, and we can have simple diffusion or facilitated diffusion. So now that we are talking a little bit about the concentration gradient and talking about diffusion and movement of molecules, we're going to talk about something called tonicity. And tonicity, the definition is the relative concentration of solutes outside of the cell relative to inside of the cell. So what we're talking about is this difference between the amount of solutes on the outside compared to the amount of solutes on the inside of the cell. And there are three different ways that we can talk about tonicity. So we have a hypertonic solution, a hypotonic solution, and an isotonic solution. So I have hypertonic twice, and that is incorrect. I will have to fix that. So our hypertonic solution is where the solution has a greater concentration of solutes than the solution inside of the cell. So hyper means more than. So more than solutes, meaning that there are more solutes on the outside of the cell, so a greater concentration on the outside than there are on the inside. And what this means is that if that molecule, if that solute is able to move into the cell through diffusion, whether it be simple diffusion or facilitated diffusion, if they are able to use diffusion to move through that plasma membrane, then they will move from that area outside of the cell to the inside of the cell. Also, what we would see is the movement of water. And this is called osmosis, which we'll actually see in the next slide. So in the case of a hypertonic solution being on the outside of the cell, so the solution that the cell is sitting in is hypertonic. This means that water will move out of the cell into the surrounding solution and that the cell is going to shrivel down. And the technical term for this shriveling up of the cell is crenation, or to crenate. So what we see is this movement in this attempt for the cell to reach equilibrium. It wants to reach equilibrium with the solution around it. And when it's trying to do this, if we take one of our blood cells, for example, and put it in a beaker of solution that is hypertonic to the cell, then what we see is either we see the solutes that are in the beaker moving into the cell if they are able to pass through the membrane, and or we will see water coming from the inside of the cell out of the cell. And so that's so it will dissolve, or it will try to dilute the solutes that are outside of the cell. And you can imagine if we take a cell and we put it in a solution that is very, very, very hypertonic, so has lots and lots and lots of solutes, many, many more solutes than the inside of the cell, then what we see happen is lots and lots and lots of water will leave the cell. And when that happens, it will get very, very, very tiny because we're losing all the water in the inside of the cell, and the cell will crenate. Notice that when I'm talking about tonicity, when I'm talking about a hypertonic solution, I have to reference that term. I have to say that the solution is something in relationship to the solution inside of the cell. So I'm saying that the solution outside of the cell is hypertonic to the solution inside of the cell. Because if you say a situation is hypertonic, for example, that does not tell a person if the solution that is inside of the cell is hypertonic, so has more solutes than the outside, or if the solution that's in the beaker or on the outside is hypertonic. So it's best to kind of reference your sentence or create your sentence with reference points, saying that the solution that the cell is in is hypertonic, meaning that it has more solutes than the cell does. So keep that in mind as you're using these different terms: hypertonic, hypotonic, and isotonic. So the next one is actually hypotonic, and I apologize for the typo there. It's a pretty poor typo. So hypo, H-Y-P-O-T-O-N-I-C. And so hypotonic means less than. Hypo meaning less than. So in this case, the solution has a lower solute concentration than the solution inside of the cell. So again, if we take our blood cell and we put it inside of a beaker, and inside of the beaker we have a hypotonic solution, that means that that solution has less solutes than the cell does, which means that we would see the opposite situation. We would see the contents that are inside of the cell, so the solutes that are inside of the cell would start to leave the cell and go into the surrounding solution if they are able to pass through the membrane. So remember, we want to reach equilibrium. The cell always wants to try to reach this equilibrium. So it's going to try to lose some of its solutes to the exterior to try to equal out the amount of solutes inside the cell and the amount of solutes outside of the cell, or the concentration of solutes. Also, what we will see is water movement. So we see water will move into the cell and it will cause the cell to swell. And so remember, the same situation, we want everything to be equal. And so if inside of the cell has a lot more solutes than outside of it, and we want to make things equal, the water will move into the cell in order to try to dilute the contents so that the inside will equal the outside. And since our cells can only hold so much water, they will swell and swell and swell and swell. And if it's an animal cell, if the solution outside of the cell is extremely hypotonic, has so little solutes in it compared to inside the cell, so much water will move into the cell that it will end up bursting. And that is called lysis. So that L-Y-S-I-S. So lysis. And so if you place, for example, one of our red blood cells into a beaker of clean water, into deionized water. Then what we see is immediately all of the cells, or that one single cell, will completely explode because so much water is moving into the cell trying to dilute the contents in the cell, and then it bursts. Then our last term for tonicity is isotonic. And so iso meaning the same. Isotonic is where the concentration of solutes is the same inside and outside of the cell, and the cell will remain the same. So in this case, we will still see solutes moving back and forth between the plasma membrane, and we will still see water moving back and forth through the plasma membrane. But there will be no net movement, which is why I have net movement in parentheses there. So there is no net movement. In hypertonic solutions, if we put a cell in a hypertonic solution, we see a net movement. We see more water moving out of the cell than we do in. So there's a net movement out of the cell. In a hypotonic solution, we see a net movement of water moving into the cell because it's trying to dilute the interior or dilute those contents. In isotonic, we still see movement. There's absolutely always movement of water in cells all of the time. However, there's no net movement. There's just this back and forth movement of water rather than it being all in one direction or mostly all in one direction. Same thing with diffusion of the solutes. So if there are any solutes that are able to move back and forth through diffusion, then we will still see that movement back and forth, but we will not see a net movement in one direction. So they will not all move out to try to make the inside and outside the same, and they will not all move in to try to make the inside and outside the same. So keep these three solutions, again, hypertonic, hypotonic, and isotonic, in mind when we're talking about different solutions and when we take a look at osmosis in just a second and take a look at cells and how they react to their exterior. All right, so now we are taking a look at osmosis. So on the left-hand side here, osmosis is a type of passive transport by which water diffuses across a membrane in order to equalize the concentration of water inside and outside of the cell. So this is exactly what we've been talking about this entire time. We've been talking about tonicity. We've been talking about this idea of water molecules moving through the plasma membrane either inside the cell or outside of the cell, trying to equalize the solutes that are in the cell and out of the cell. So the direction of osmosis is determined by the total amount of solutes on either side of the membrane. And so water will always move toward the side that has the greater concentration of solutes. So this is saying that water will always move in the area where there's more solutes so it can dilute those solutes. So here we see our hypertonic solution. So our solute concentrations are higher in the extracellular fluid. So you see here we have all of these solutes, very, very, very concentrated solution, and they're not that many solutes inside of our red blood cell or animal cell, not that many solutes inside of our plant cell either, the same little tiny dots here. So we see our net movement of water, or our osmosis, will move in this direction, will move out of the cell. And then we see this crenation of the cell, or the shrinking of the cell. When we talk about animal cells, we see that the entire cell will shrink down, and actually you can see that it becomes spiky. And so that entire thing shrinks down and becomes spiky. In the case of the plant cell, however, they still have that very nice solid cell wall surrounding the cell. And so what we see is even though we have this water movement out of the cell in plant cells, we see the plasma membrane shrink down very, very, very small, just like the animal cell does, but the cell wall stays intact. So you can see they kind of show here that the plasma membrane is getting spiky, so it's shrinking down, getting spiky, but our cell wall still stays intact. So we still kind of maintain our nice firm cell wall but we are losing a lot of water in that case. Next, we see our hypotonic solution. So the solute concentrations are lower in the extracellular fluid, and therefore water diffuses into the cells. So here we see less solutes on the outside compared to the inside. We see a lot more solutes in here and a lot more solutes in here. So osmosis will move in this direction. So we have water movement into the cells trying to dilute all of these solutes. And in the case of the animal cell, or the red blood cell, as I mentioned before, we have lysis. So that cell will actually burst because so much water is trying to dilute the contents of the cell. On the left-hand side there, we see the plant cell. And again, the plant cell has this protective function because it has the cell wall. So we have this net movement of water into the cell, diluting the contents of the cell. However, it can only fill up so much because that plasma membrane, even though it will take in as much water as it possibly can only do that until it reaches the very edges of the cell wall. And once it's completely filled up that interior compartment, you can see it even kind of got a little bit larger than we saw before. It kind of puffs out a little bit. However, it does not lyse. So the cell does not actually burst because it has that cell wall protecting it. Then lastly, we have our isotonic solution. And so we have our solute concentrations are balanced. So about the same amount of solutes are inside the cell as they are outside of the cell. And so you can see the arrows go both ways. They go out of the cell and in the cell because water is moving back and forth here. And so we do not have a net water movement, but we always have water movement in osmosis or in cells. On the right-hand side here, it kind of mentions celery. So it's kind of at a take-home message here, is that if we take our celery and we place it in distilled water, so water that has very, very few solutes in it, so not a whole bunch of solutes, that would be a hypotonic solution compared to the cells. Then we see here it mentions the distilled water contains fewer dissolved molecules than the celery cells. Water molecules diffuse into the celery, equalizing the water concentration inside and outside the cells. And so the celery becomes crisp. So if you ever want to crisp up your celery, if it becomes wilted, you just need to put it in some very clean water, some distilled water. Then we see this situation on the left-hand side happen where all of the water will move into the cell, trying to dilute the contents, and you'll have very crisp celery. On the right-hand side there, you see that we can place that same stalk of celery in salt water, very salty water. So this would be a hypertonic solution. And so we see the water movement moving from inside of the cell to the outside of the cell, trying to dilute all of the salt in the water, and then our celery becomes wilted because our plasma membrane shrinks and the cell is much, much smaller, so it's dehydrated, and so it wilts. And you can see up at the top our dehydrated celery contains many dissolved molecules relative to water molecules. So we have our water has moved outside of the cell trying to dilute the exterior, leaving a very, very small plasma membrane behind with lots of solutes. So the direction of osmosis is determined only by a difference in total concentration of all the molecules dissolved in the water. So we've been talking about this idea of an area of high concentration of solutes and low concentration of solutes. When we talk about concentration of solutes, it does not matter what solutes they are. It's the total concentration. So if we took sugar water and salt water and mixed them and we put red dye in it and other dyes and anything else you can think of, if you put it all in the same solution, you have all these different types of solutes, and then you take a celery stalk and put it in there, then we would still see osmosis occurring in the way that we would see it moving out of the cell. We would see water moving out of the cell to try to dilute the contents that are in the beaker. And that is due to all of the molecules that were put in there. So the sugar, the salt, the different colored dyes, anything else we put in there. So it's the total concentration of the molecules, not just one single one affecting osmosis. So it does not matter what solutes they are. So those were our kind of 3 types of passive transport, which were kind of 2. So diffusion and osmosis are passive transport types, meaning they do not require the use of energy. Our first one, diffusion, can be broken down further into simple diffusion and facilitated diffusion. So simple diffusion meaning it simply goes through the plasma membrane, does not need any help, and facilitated diffusion is that it is facilitated, or a protein facilitates the movement of the molecules. But again, it does not take energy. Now we're going to take a look at a lot of different examples of active transport. So molecules cannot always move spontaneously and effortlessly in and out of cells, just as we were talking about with diffusion and osmosis. So cells use energy to move small molecules, and there are 2 different types that we'll talk about first. We'll talk about primary transport, or primary active transport, and secondary active transport. And so these 2, as is mentioned, differ only in the source of the fuel, or the source of the energy, and we'll see how that is coming up. So our primary active transport uses energy directly from ATP. So it's right from the source. It's primary. It's first. Using ATP as it is right there. So active transport occurs when the movement of molecules into and out of a cell requires the input of energy. And again, this is when we're moving from an area of low concentration of solutes to high concentration of solutes. So sometimes we want things to go in the opposite direction, to where we really, really want to create this huge difference in our concentration of solutes. And so even though we have less solutes in one area, either in or out of the cell, we still want to move whatever solutes are in there to the opposite side where there are more solutes. And that's when we have to use energy, because it does not just happen spontaneously. So here you can see, for example, in response to eating, the cells lining your stomach use ATP to pump large numbers of hydrogen ions into the stomach. So here is inside a cell lining of the stomach. Here is outside the cell and inside of the stomach. So here we've just eaten something. We have a high concentration of molecules here. Inside of the cell lining of the stomach, we have a low concentration of molecules here. But what we want to do is we want to take the hydrogen ions and put them into the stomach to help try to break down the food items that we've just eaten. So we use one of our embedded proteins, or integral proteins here, and it requires ATP because we're talking about active transport. So you can see that that molecule, that protein itself, uses the ATP in order to shift one of those hydrogen ions from the area of low concentration to an area of high concentration. And so what it kind of does is acts as a swinging door that requires energy. So the door is open to one side. You can see on the left side the door is open to this side, and then once the hydrogen molecule goes in, we use up our ATP, and then it opens the other side and allows it to go through. And so this is primary active transport. So again, primary meaning first or at the source. So it uses ATP right on that protein molecule that's shifting the hydrogen ions from an area of low concentration to an area of high concentration. Then we have our secondary active transport. And this is where ATP is not used directly, it's used indirectly. So that's why it's secondary. This is a situation where we move one molecule against its concentration gradient, so an area of low concentration to an area of high concentration, while letting another one flow down its concentration gradient. So moving from an area of high concentration to an area of low. So we still are using ATP, we still have to use energy, But what happens is there's energy, or right here, the energy from ATP was used to pump one of the types of molecules involved against their concentration gradient. So it's not used directly. We're having two things move. We are allowing one to flow down from an area of high concentration to low concentration, and then using energy indirectly, using the energy that creates to move molecules against their concentration. So moving from an area of low to high. And so this is not a direct relationship where the protein is just able to use the energy, take it from against its concentration gradient. But in this case, it's necessary to allow one solute to move down its concentration gradient, and then that allows us to use that energy to move the other one against its concentration gradient. So make sure that you understand the differences between our primary active transport and our secondary active transport and how that is. So another type of active transport or transport that uses energy can be summed up as bulk transport. So when we are talking about our primary and secondary active transport, we are still talking about relatively small molecules, things that can move through these little protein channels. When we talk about bulk transport, we're talking about very, very large molecules. And so these molecules are much too big to get into a cell through passive or our other active transports, our primary transport or secondary transport. And so we have to use what is called endocytosis or exocytosis. So these two, endocytosis and exocytosis, are used for these bulk transport. So these very, very large molecules that we cannot get into the cell or out of the cell otherwise. So we'll be taking a look at phagocytosis, pinocytosis, and receptor-mediated endocytosis. Those are three different types of endocytosis. Okay, and then there's just exocytosis by itself. So let's take a look at that. So the first one is phagocytosis. Phago means eat, and cyto means cellular. So this means cellular eating. So we're taking these really large particles, and the cell will gobble them up, or will engulf and eat them. This is a type of endocytosis. Endo meaning into. I remember endo is into because they both have ends. So this is something going into the cell. So endocytosis, one example is phagocytosis. So phagocytosis is a type of endocytosis by which cells engulf large particles. You can see here that they show on the bottom in the illustration the plasma membrane here on the outside of the cell, and they show the very large molecule. What happens is we have this invagination, which we've mentioned when we talked about the endosymbiotic theory. This invagination is where we get this dip in the plasma membrane. So we have this dip in the plasma membrane that kind of sucks in this large particle. You can see it continues down here. So we kind of start to dip in, and then it continues to dip inside of the cell and engulf this this large particle here, this molecule. So the plasma membrane forms a pocket-like vesicle around a large particle. So a vesicle, for a definition, is this kind of glob, this little bubble, for example, that moves around the cell, and it allows things to be transported. So you can see here that we have this invagination, or this kind of dipping in of the plasma membrane. Then it creates this pocket. Once it creates this pocket, then it will pinch off that pocket and get it into the cell. So you can see that the top part of the plasma membrane here, what will happen is this side will then merge with this side of the plasma membrane, and it will just pinch off that entire pocket, and that becomes a vesicle. And so vesicles are extremely important for transport. You can see here that it can engulf a particle and use that vesicle to transport it inside of the cell so that it can then degrade or break down that product. We also use vesicles in transporting things throughout the cell. So when we're moving from organelle to organelle to organelle, we can use these little bubbles or pockets and move them throughout the cell. So we're able to transport them. And we can also use them to transport something outside of the cell, which we'll see in just a moment. So phagocytosis is cellular eating. You can see up at the top there an actual cell is going to eat up this other cell or molecule on the left. And so cellular eating, or taking something very big and bringing it into the cell. Then we have pinocytosis. Pino means drink, and cyto, again, means cell. So this is cellular drinking. And in cellular drinking, we see that we bring in large molecules, but we kind of drink, as it is in the term, to where we're bringing in a whole bunch of that fluid in with us. So we're able to pull in a whole bunch of other molecules with us. So we see the same type of invagination where we actually, in this case in pinocytosis, they're describing that there are receptors, and that the molecules will attach to those receptors, and then it will start to invaginate, just as we saw with phagocytosis. However, in phagocytosis, I should back up just a moment, you can see in this cell at the top, that they create these kind of little arms that go around. So in phagocytosis, we create what are called pseudopods, or fake feet. These pseudopods act as little arms that kind of grab onto that molecule and pull it into the cell. And that's what's a little bit different between phagocytosis and pinocytosis. In pinocytosis, we kind of suck in whatever fluid comes with those molecules. In phagocytosis, we kind of actively have these lumps of cell that come out, act as little arms or false feet, and grab onto that molecule. So there's the key difference between those two types of endocytosis. Again, endo is moving something into the cell. So we see here that we have our coated pit. So we create that pit, or we create that kind of bubble, and then we bring it into the cell as a vesicle. And you can see on the right-hand side there's an actual picture of pinocytosis occurring inside of a cell. Then our third type of endocytosis is called receptor-mediated endocytosis, which is similar to the picture we just saw with pinocytosis, but is more specific. So receptor-mediated endocytosis is a type of endocytosis by which cells engulf specific particles. So what we see here is a specific particle. The example here is our LDL particle. And on the outside it has these cellular markers. Remember, we mentioned that proteins, or the glycocalyx, is used as a cellular marker. And so we have these cellular markers that will attach onto these receptors in one of our cells. So this is saying in a liver cell. So in the liver cell, we have our individual proteins that are embedded in the plasma membrane, and those are our receptor cells. And then our LDL particle has proteins sticking out of it also, and those attach to those receptors on the other cell, on the liver cell. So here, step one, LDL binds to the receptor protein that reside in the plasma membrane of the liver cells. So kind of like that key in the lock, we have the key on the LDL particle, fits in the lock on the receptor protein on the liver cell. Then the plasma membrane forms a vesicle and engulfs that LDL particle. And then once it engulfs the LDL particle, it does the same thing, it invaginates, which means it creates this pocket, and it dips down into the cell, and it pinches it off, and then we have our vesicle. So then LDL is broken down into useful molecules such as estrogen and testosterone. So what happens is, in endocytosis, we saw phagocytosis, which is gobbling up or eating, cellular eating of large molecules or large particles. This can also be bacteria or a virus, for example. Our cells will seek out a bacteria if it sees that it's not supposed to be in the body, and it will use phagocytosis to grab onto that bacteria and suck it into the body, or suck it into the cell. when it does that, it's inside of this protective vesicle. And as is mentioned here, we take that vesicle and then our body, our cell, can use lysosomes, which we'll see, to break down that particle. So we'll actually bring it into the cell, and then it will merge with some enzymes that will break down that bacteria and kill it for us. So kind of the goal of endocytosis here is to bring something into the cell so the cell can use it, or so that the cell can break it down and kill it if it's a bacteria or a virus, for example. Then lastly, we have our exocytosis. So exocytosis is the method by which cells export products for use in another location. So this is exiting the cell. Exocytosis is exiting the cell. So we can see here that it kind of happens just the opposite that endocytosis does. We have our molecules that we want to export, or that we want to exit the cell here in our transport vesicle inside of the cell. Then we use our transport vesicle, bring it up to the edge of the cell here, and all of our vesicles are made up of the exact same stuff as our plasma membrane, because we pinch it off to bring it into the cell. So in this case, we're going to allow our vesicle to merge with the plasma membrane, and in that case, it will then force that stuff out of the cell. So 1, our molecules are packaged in a vesicle within a cell. 2, the vesicle fuses with the cell's plasma membrane, and then the vesicle contents are released for use throughout the body. So they'll go into the bloodstream and be brought to where they need to be. So we can see this in hormones, for example. When we make hormones, we make them in one of the organs in our body, but then they need to go elsewhere in order to function. So we package them up in our transport vesicle, bring them to the edge of the cell, and dump them out into the bloodstream so that they can travel to other parts of the cell where they're needed. So this is exocytosis, which is exiting the cell. So something for exiting the cell. All right, so those are our types of transport, how we get things into the cell and how we get things out of the cell. And now we're going to talk about how cells can communicate. So different cells have cell junctions. Cell junctions are connections between cells that hold them in place and enable them to communicate with each other. So some cell junctions are specific only to hold cells in place. Other ones allow cells to communicate very, very quickly with each other, and we'll see the differences in those. So some of the cell junctions that we will look at are tight junctions, desmosomes, gap junctions, and Plasmodesmata, which are only found in plant cells. So all of these cell junctions involve numerous types of protein and glycoprotein adhesion molecules, so things that are adhering or sticking two cells together, and we'll take a look at how that works. Alright, so first up are our tight junctions. So our tight junctions form continuous watertight seals around cells and also anchor cells in place. So just as their name implies, they're very tight. They are kind of like, as you can see in this picture over here, what I liken them to is taking a needle and thread and sewing the two cells together. You can kind of see this netting or this webbing between the two cells. And even at the bottom here, on the actual picture of a tight junction, you can see how kind of there's this big thick wrapping in that area. And so you can see kind of the sewing between the two cells. So these actually form a watertight seal between cells, like caulking around a tub. So it's extremely tight, so water cannot move between these two cells. And the cells are closely stuck together, and so even if you tried to pull the two apart, it would actually tear the cells because they're such tight junctions. So that's their anchoring function. So they're particularly important in the small intestine where digestion occurs. So if you can imagine, our intestines are always moving back and forth and around, and all of these cells are being moved around all of the time to try to break down our food and get it into the cells. And if we always have those cells moving back and forth and crunching up food, you can imagine they'd get a lot of tear, a lot of wear and tear. And so in order to not allow food particles to go directly into the bloodstream where that could become dangerous and actually kill somebody, instead we have these very tight junctions in between the two cells so that as they're moving around, there's no movement between the two cells so that nothing is able to get through. So we're not actually getting food particles into the bloodstream. We have to break them down before we get them into the bloodstream. So that would be, for example, if you decided to take a needle and put a ground up potato chip into your bloodstream. That would be very, very dangerous. So our tight junctions are there to prevent big food particles like that from getting into the bloodstream. So it holds the cells very, very close together, even though they're moving around a lot. Next, we have our desmosomes. And so desmosomes act as spot welds or as rivets that fasten cells together. So that's what I most often think of them as. You can see here they have this kind of chunk of stuff, which is actually called plaque, not the bad plaque, but this kind of layer of plaque here that acts as a rivet. So there's a layer of plaque on one cell, a layer of plaque on the other cell, and then we have these proteins that are embedded inside of that plaque that stick between the plasma membranes. And so it allows them to kind of act like Velcro, as is mentioned here, or spot welds, to where we have these spots that are just right here in this area. One single desmosome, little tiny spot welds that act kind of like Velcro. So you can see that they're stuck together. You can see they are held very close together, but they're not like the tight junctions in that they're so close together that they do not allow things to get through. So in this case, they're not going to allow large particles to get through, but they are not watertight. So you can get small particles and water to get through those two cells. And so we find this, or these desmosome junctions, or cell junctions, in much of the tissue lining cavities of animal bodies. So we still want fluid to get between our cavities, so lining the cavities of our bodies, but we certainly do not want to have large particles moving through. So these will kind of prevent large particles from moving through while allowing small particles and water, but at the same time it's holding the cells together. So we still want these cells to be held together, otherwise, of course, our tissues would be falling apart all the time, but we still want to have some fluid movement between them. So that's why we use desmosomes in some places. So we have tight junctions that do not allow anything through, desmosomes which will allow fluid through, and then we have our gap junctions. And gap junctions allow lots of things through. So gap junctions act like secret passageways between, and allow materials to pass between cells. So you can see here that they are kind of like pores. We have a tunnel between the two cells. So here's one cell, here's the other cell. And so they're pores surrounded by special proteins that form open channels. So you can see there's one protein here, there's another protein, there's another protein. We have all of these little tiny, long tubular proteins that all come together to make a pore. And so they allow for this kind of tunnel to be created. So this isn't so much about water flowing in between the cells or molecules flowing in between the cells, as in on the outside of the cells. But what it is it's allowing molecules to move from the inside of one cell to the inside of the other cell. And these are important mechanisms for cell-to-cell communication. So you can see down here, this cell has lots and lots and lots of gap junctions, all these little tiny tunnels between this cell that we're kind of in this picture, and the other cell that we would be going to. Some of the places that we find so many gap junctions like this would be inside of our heart. So all of our heart muscle cells have these gap junctions because we need this electrical impulse and we need all of these ions to move very, very quickly through the muscle cells in our heart so that we can contract all of them at the same time when we pump our heart. So gap junctions are extremely important, specifically in our heart muscle or in the cells in our heart, so that we can get everything very quickly through these cells, going right through these tunnels that allow the passage of these molecules. Lastly, we have our plasmodesmata. And plasmodesmata are basically like gap junctions that we find inside of plants. So these are tube-like channels connecting the cells to each other and enabling communication and transport between them. So you can see over here that each one of these are individual plant cells. They have their cell walls on the outside. And then we kind of zoom in on one spot on the cells. So here the green is the cell wall, or two cell walls right next to each other, the two cells. And then we have the plasma membrane on either side. So again, we still have the plasma membrane on the inside of the cell wall of the plant cell, on the top cell and the bottom cell. But then we have these tunnels. So similar to the gap junctions, we have tunnels that allow quick movement of molecules between the cells. So it allows things to move between the cells, and so basically it kind of creates this idea that plants are all kind of one cell because they have all of these plasmodesmata in them, which allows the water to move from cell to cell to cell. So when we water a plant from the bottom, we can pull the water through each of these cells, through the plasmodesmata, as well as the nutrients that are in the soil. So as the plant is gathering the nutrients from the soil, from its roots, it can transfer those from cell to cell to cell up through the entire plant. And so then we end up having this quick movement throughout the plant so that every single cell in the plant is able to get all the molecules and water that it needs. So these plasmodesmata are extremely important for that function. So gap junctions and plasmodesmata are extremely similar, except we find gap junctions in animal cells and plasmodesmata in plant cells. And remember then we have the two other types we discussed, which were tight junctions, meaning the ones that are kind of like sewing two cells together, stitching two cells together, very tight, does not allow for passage. And then our desmosomes, which are kind of like spot welds or rivets that hold cells together, but they do allow the passage of fluid.